In this video series, we're going to explore some more exceptions to Mendelian genetics. We're going to focus on the concept of multiple alleles. This will be a series of videos, 2 or 3 parts, where we're going to go over some concepts and do some practice problems together. So remember that Mendel did some experiments with peas and found these ratios, these 3-to-1, 1-to-1, 9-3-2-1 ratios. We didn't quite see this yet, but we'll see it eventually. But this is a list of the assumptions he was making. And the last one we saw was that he assumed that alleles will show complete dominance. So basically big R will be expressed if little r is there. But we saw that that's not necessarily the case. Sometimes they blend and we'll see incomplete dominance and we'll see codominance. Today, though, we're going to focus on another assumption that we have in Mendelian genetics. It's the idea that for every gene, there's only 2 alleles. So for example, for flower color, you might have red allele and white allele, and that's it. There's only sort of 2 varieties. So we'll find out that this is actually not the case. So let's go over here. We'll take a look at this little prompt before we move on anymore. So below we have some flowers. We need to identify their phenotypes and their genotypes for each one of them. And so you've already seen this example in the Mendelian genetics lesson, but basically purple was dominant to white. And so let's say we used big P for purple and little p for recessive. Well, this would be little p over here, and this could be big P, or it could be big P. We're not sure which one it is, but it could be any one of those, and we're assuming complete dominance here, okay? Now, in this scenario, we only have 2 alleles. We have the purple allele and the white allele, 2 versions of that gene. In reality, sometimes that's not really true. Sometimes you might have more than 2 different versions of a gene. Let's take a look over here. This picture here is a picture of different blood types. So you can see these are the different, these are red blood cells over here, the cells that carry oxygen in your body. And I want you to take a look and try to notice what's different in them. What looks different between this blood type, this one, and this one. So think about that for a minute. Look at the picture, just spot the differences. And what you'll see is that what's different is really the shape of this structure that's on the red blood cells. So here we have triangles, here we have circles, here we have triangles and circles, and here we have nothing. So these are some different phenotypes that you could have for red blood cells. And those different phenotypes, they're basically the different types of blood that are out there. Type A, type B, type AB, type O. You might know yours, you might not know yours. You could inquire about it, and there's tests you can do to figure it out. But for now, just understand that we have all these different blood types here. So that's interesting. Think about this carefully. Let's come up with some genotypes for these with letters. And you'll notice that you might actually find this quite challenging to do. Up here it was pretty easy. There's 2 versions of the allele, big P, little p. So this is little p, and this is, well, little p, big P, and big P. But that only gave us 2 different possible phenotypes. You'll see over here there's way more than 2 phenotypes. There's 1, 2, 3, 4 different phenotypes, which is weird because you'd think there'd only be maybe 2 possible ones. If there's incomplete and codominant, maybe 3 possible phenotypes. But there's more than we can explain if there's only 2 different versions of a gene. So that brings us to the exception of what blood types are. Blood types go against the assumption that there's only 2 alleles. Sometimes you might have more than 2 alleles, and so we call that exception multiple alleles. So Mendel assumed that there's only 2 alleles for every gene. So for example, for plant height, the 2 alleles are big T and little t, tall and short. But for many characters, there's actually multiple alleles. And so when you have multiple alleles, that usually leads to more possible phenotypes, like we saw with the blood types A, AB, B, O. So ABO blood types are an example of a character in humans that have actually 3 alleles, 3 different versions of the blood type gene. And so we call it IA, IB, and I. And depending on the combination, you always get 2 regardless, okay? You only get 2 of these. Depending on the combination you get, you'll be a certain blood type. So this combination that you get will determine what carbohydrate is on your red blood cell. And those different shapes we saw in the red blood cell, those are carbohydrates. Basically special sugars that determine how your blood kind of reacts to certain things. So you can see that there's 3 possible alleles, IA, IB, I. IA usually makes the triangle carbohydrate. IB makes the circle carbohydrate. Doesn't really matter. It just makes a different carbohydrate than A. And then little I does not make any carbohydrate. So let's go see some possible combinations. You'll see over here that if you get IAIA or IAI, you get type A blood, triangles. So that means that IA is dominant to I. If you get IBIB or IBI, you get type B blood, the little circles on there. Again, meaning that IB is dominant to little I. If you get IAIB, you get both. Circles and triangles, you get type AB blood. They're expressed together. Interesting. We have a bit of codominance happening here too. And if you got little I, you don't get any carbohydrates, you're type O. So little I is definitely recessive to all the rest, but IA and IB are codominant to each other. So you have a bit of everything here. You have multiple alleles, meaning you have 3 different versions of a gene possible. You have some codominance happening here, IA and IB, but you also have complete dominance when you have IA paired with I or IB paired with I. So for type A, you'll notice you could be homozygous A or heterozygous A. For type B, you can be homozygous B or heterozygous B. For type AB, you're simply AB. And for type O, you're simply II, homozygous recessive II. Okay, so blood types are certainly an exception to Mendelian genetics in a few different ways. The main one is that we have multiple alleles, not just 2 versions like big P, little p. The other way is we have that codominance aspect. So do you know your blood type? If you do, think about it. If you don't know it, or if you do, why should you care? So when you're looking through the slide deck, take a look at the video that's included here so you can get an idea as to why you should care about your blood type. Some people donate blood and that saves lives. And there's certain blood types that are in demand because we don't have enough of it for people who need blood to survive. So watch that video and understand why it's important to know your blood type. So that first half that we just covered was basically just a little bit of an introduction to the concept of multiple alleles, blood type being an example of a trait, a character that has multiple alleles rather than just 2. In the next video, we're going to do some practice problems that go over blood types.